And thank you very much for Liana and Giovanni and Chris for this wonderful opportunity to present my work with you today. I will speak today on antiviral activity of glucosylceramide synthase against viral infections. So I started to work on viruses before no one knew what viruses, like before the COVID. And before the COVID, actually, it's also nice for me to recall what I did in my previous life. It's the pre-COVID and the post-COVID life. So when I started my work on viruses, I was most interested in a virus which affect the brain, the central nervous systems, and in a viral encephalitis. When virus enter the brain, it leads to inflammation of the brain parenchyma. And the symptoms can be varied. It can be flu-like symptoms, very mild disease, but it can also complicate to more serious and even a chronic, long-lasting neurological impairment. So I will just want to present you a few viruses, which I will talk about them in the lecture. I will also discuss in the end about the SARS-CoV-2, of course. When we speak about a viral encephalitis, and that was my interest, there are a few families of viruses, which I will focus today mostly on synthase virus. Synthase virus is from the alpha virus family, the same family of the EE and WEE, and we use this as a model that we can use in build 2 level, so it's more easy to work in the lab. And also about the West Nile virus, both viruses are used as a model for viral encephalitis. So why to study Sphingolipid and viruses? What is the connection? What is the rationale behind it? So this is a general overview of the life cycle of a virus in the cell. So as we can see that in every step in the life cycle of the virus, it uses a membrane. If it's in the attachment, so it's the receptor can be lipid or it's inside the membrane. So the membrane properties can influence about the attachment and entry of the virus. The entry is by endocytosis, which of course uses the membrane machinery of the cells. Also, when we speak about replication of the virus inside the cell, many of the viruses using the replication factory, which they create, which are membrane embedded. And of course, when we speak about envelope viruses, when it go outside the cell, the virus don't have its own membrane, but it just take a piece of the membrane of the host. So every difference in the host membrane can influence of the mature virion which leave the cells. Beside the rule of the life cycle of the virus, Sphingolipid is an important role in other biological process, which is very relevant to viral diseases. Cell signaling, cellular process such as apoptotic, and very important thing is the immune system. And the immune system is one of the crucial host defense and crucial mechanism that is responsible for and play a very significant role in infectious disease in the outcome of it. So the first question I ask when I start is, the assumption was that if Sphingolipids are so important for viruses, so does viruses can influence this pathway or the Sphingolipid level upon infection? So Okay. So I'm sure that you are all familiar with this, the metabolic pathway. Every sphingolipid is a synthase and hydrolyze. So the first experiment we did, we put NBD sphingonin, which is the precursor of the sphingolipid and metabolic pathway. We put it on cell 3 hours post-infection. Now, most of the experiment that I will talk about them, we were interested in the early time point after infection. Like there are in the literature, there are many works showing differences in lipid after 24 hours, 2 days, but this is also, it can be a very downstream effect, but not a direct effect. So we were interested in what happened just at the beginning of the infection. So 3 hours post-infection, we put NBD sphingonin only for 10 minutes, and we looked for its incorporation. So this is an HPLC that we did. This is the sphingonin peak. The black line is the control, the uninfected cells. And you can see that most of the sphingonin is still remains as sphingonin, and only part of it converted into other sphingolipid. And this is in the infected cells, we see a decrease in sphingonin, meaning it gets into the pathway, and we see this peak that we have more sphingolipid that were generated. So we also send this sample for mass spec analysis, hoping to see which is this peak, what the virus, which sphingolipid the virus induce or elevate early upon infection. In these cells, in virus cells, we only see what we saw here. We saw the decrease in sphingonin and sphingosine, but we couldn't detect which sphingolipid is the upregulation, and this is probably to the limitation of the method. So we didn't have an answer to which lipid is the end product of this induction of sphingolipid pathways. So we went to in vivo model. In this model, we infected mice with synthase virus, and we detected the lipid profile in the serum 5 days post-infection. Now, the reason we choose serum and not brain and other organ, because the lipid composition can be changed via the cell component. So if, for example, in the brain you have more inflammation, you will have more inflammatory cell, and this can be influenced about the sphingolipid. So we checked in the serum, and we were very surprised to see elevation of ganglioside, GA1 and GM2 in the serum. And this fit with our assumption that the synthase virus induced both in vivo and in vitro alteration in sphingolipid upon infection. So we hypothesized that the virus, when it enters, induces this pathway, like increase synthesis, de novo synthesis of sphingolipid, and leads eventually to accumulation or upregulation of certain glycosphingolipid, as we saw the GM1, for example. Now, if we see accumulation of glycosphingolipidin is a phenomenon that we also see not in viral infection, but also in lysosomal storage disorder. That was my field that I work in Tony's lab, mostly about Gaucher disease. Gaucher disease, for example, caused by a mutation in the gene encoding glucosylceramide, leading to accumulation of glucosylceramide. So we see here two different etiologies of disease and two different. In viral infection, we believe that the reason for elevation of sphingolipid is due to increased synthesis. And in lysosomal storage disorder, the elevation is because decrease of degradation, But no matter what the reason, the outcome in both scenarios is the same. And the outcome is more sphingolipid or glycosphingolipid in the patient. So then we asked, so can we take a therapy that was designed to treat lysosomal storage disorder and use it for a viral disease? Because in both scenarios, we want to decrease the level of accumulated glycosphingolipid. So we reached to Genzyme Sanofi, knowing that they have glucosylceramide synthase inhibitor. And the reason we approached them, we wanted to have, because our interest was in encephalitis and viral infection of the CNS, so we wanted an inhibitor which crossed the BBB. So GZ161 crossed the BBB, and they also gave us the GZ346. This does not cross the BBB. They're, as much as they can tell, they're specific to glucosylceramide synthase, because other inhibitor like the NBDNJ also regulate other enzymes. It's not that specific, so it was important for us. So we took this inhibitor, and first we tested in vitro on Vero cell, same cell that we saw that lead to this decrease in sphingolipid and sphingosine, and test whether it's inhibit the replication of the virus. And as you can see, both GZ161 and 346 inhibit this is percent of inhibition, inhibit the virus replication. This is one of the non-structural protein 2, one of the early, this is the most early protein that is being translated upon a viral infection. So we see that both of them inhibit the translation of this protein. So we realized that the synthesis of glucosylceramide, that it's not necessarily glucosylceramide, but glycosphingolipid. So inhibition of glycosphingolipid synthesis. This synthesis is important for the viral replication, and that can explain why the virus induces it upon infection, because it probably needed it for its replication. So then we wanted to test it in vivo. So we test two regimes. One, when we treat the mice 5 days before infection, when we spoke to a Genzyme with Sanofi, so they recommended on this regimen that you have the most efficiency reduction in glycosphingolipid. And also, but this is cannot be as a treatment, because treatment you always start after you got the infection. So we also tried 2 days post-infection. I would just say that the mice died at day 5, so it's the therapeutic window is very small in this severe model. So first, as I showed you before, the SVNI leads to elevation in the serum of this mice, so GA1 and GM1. And when we treat the mice with GZ161, it really did what it's supposed to do, inhibit the synthesis of glucosylceramide, and it's reflected here by the reduction. There is no elevation of a ganglioside in the serum of the mice when we treat them with the inhibitor. This is a survival curve of the mice. So mice which get no treatment, and 90% of them died. When we treat the mice, this blue line is 5 days before, it is the optimal treatment. We have a 60% survival of the mouse. We increase the survival rate, and also when we start to treat 2 days after the infection, we see a beneficial effect of the drug. So it didn't rescue all the mice, but is extend the survival rate of the mice. So we wanted to see what is the mechanism by which GZ extends the survival of the mice in vivo. Now, the most simple explanation that we thought, and that would be because we saw that it inhibits virus replication, we were sure that we'll see much less virus, and the simple explanation would be we have less virus, and the body can then cope with the smaller amount of virus, and that would be it. And we was very surprised to see that when we checked the reduction in viral load in the brain, that we didn't see any significant reduce. Now, I didn't mention it in the in vitro assay, but maybe I will just go back to here. So you can see here that also in the in vitro, although it inhibits the replication and it's significant, in the range of its non-toxicity, we can reach to 50% inhibition, which in terms of viruses, it is not that high, because you can see here in vivo, this is half of the amount of viruses. So instead of have 5 to the 7 viruses, we have 2.5 to the 7. It's not that dramatic. And actually this reciprocate the in vitro, but this cannot explain this beneficial effect that we see in vivo. So then we know that the virus, the death of the neurons upon a viral infection is apoptotic. So we measure the same tissue that show no difference in viral load. We measure the Caspase-3-7 activity to see if they have a difference in neuronal loss, in neuronal pathology. And we saw a significant decrease with GZ of the neuronal loss. So although we have same amount of virus, we have less neuronal cell death. So we look for the mechanism. Yes, it doesn't. Okay. So in order to look for a mechanism that can explain this, we did a transcriptome analysis of this brain. This is the SBNI viruses, and you see that this is the genes that the treatment with GZ, they didn't go up. And if we look on this gene and see the list, these are the go terms of them, we see many or all of them related to the inflammatory reaction, and most of them to the adaptive immunity, T cell, NKT cell, iNKT cell, etc. So we assume that the difference is related to inflammation and to inflammatory cells. And so we verified this result by FACS analysis of the brain. And indeed, we saw less. Now, we picked two groups. As I showed you in the lifespan curve, 60% of them are rescued, but not all of them. So we choose mice which weren't rescued by the treatment and mice which were rescued. And we saw that a decrease in iNKT cell, the CD4, CD8, and macrophage. So there is less recruitment of inflammatory cells into the brain, although we have pretty same amount of virus in the brain. the GZ reduced the recruitment of cells to this. So we will look for what can explain what GZ can do to impact on the recruitment of the cell, of the inflammatory response that much. And searching the literature, we saw that one of the immune mechanisms that glycosphingolipid participate is activation of iNKT cells. Antigen-presenting cells can present a lipid, galactosylceramide, and we also found in the literature that it's also a glucosylceramide and also other lipid. And while they present this lipid, they activate the iNKT, and iNKT are then can activate the other and enhance the adaptive immune response. So our hypothesis was that we wanted to check is, we know that the virus elevate glycosphingolipid upon infection, and we inhibit this elevation by GZ161. Maybe, or could, this elevation that it's triggered by the virus can trigger iNKT cells, which later on enhance or induce the adaptive immune response, the CD4 and B cells. And now galactosylceramide is a known substrate for iNKT. So what we did, we infected the mice, we tweaked them with GZ161 to inhibit this elevation by the virus, but we added galactosylceramide to induce iNKT activation. And we wanted to see that if by that we abolish the rescue of GZ161. And indeed, in this experiment, the green line is the untreated mice, and the blue line is the mice that treated with GZ161. But when we added to the 161 also GALSER, means we treated them, but we also induce the iNKT, we abolish the beneficial effect of GZ161. So we think that this is now work in progress, and we proceed to verifying this. But we think that this can explain, if I will just summarize this part, the virus induce elevation in glycosphingolipid. This elevation is necessary for viral replication, but at the same time, this elevation also trigger the host immune response. Now, I didn't show it today, but this is a published paper of work that they did in Tony Futterman, and this is something that his lab is looking at, that sphingolipid trigger interferon and the antiviral response. And it makes sense now if really this is some of the pathway that the virus induce upon its infection. So it makes sense that the host immune response evolved in such a way that it's also induced the host response, the immune response, which is antiviral. And if we speak about the therapeutic effect that we see of GZ161, I personally think that it doesn't relate to the viral replication of the virus, because it's not that dramatic, this effect. But the effect of GZ161 can be due to this alteration in the host immune response, which we know that in infectious disease can be very detrimental and have a very severe outcome. And so this is some of the things that we are now exploring. So we saw this on synthase, and we said, okay, so this is only one virus, which is encephalitic. is it something which is common to more viruses? So we also checked it on the West Nile virus and neurotropic flavivirus from a different family. I don't show you here everything, but we saw also same in vitro, it inhibits the replication of the virus in vitro, and we also see a beneficial effect, although in a lesser extent compared to the SB9, but we also see beneficial effect and in vivo. So then the SARS-CoV-2 emerged to our life while we were in the middle of working on encephalitis virus. The first things we thought, okay, we have a compound that inhibits two different viruses from two different families. So what about SARS-CoV-2? Would it inhibit or get influence about SARS-CoV-2 also? So we immediately examined that. This is the plate showing the plaque forming unit. Every dot in this plate represent live virus colony-like. It's a plaque forming unit. And this is the dilution of the plate. So when we don't treat the cells, we have many of the virus replicate in the dish. But when we treat with GZ, both of them inhibit this replication. And if in the synthase virus and in the West Nile virus, we only see 50% inhibition in the untoxic zone. So here we manage to get to more than 90% inhibition. The inhibition was much more prominent, as you can see here in this plate. So we also wanted to see whether SARS-CoV-2 also alter the Sphingolipid pathway. So again, we took bear cell, we infected them in 3 hours post-infection with SARS-CoV-2. We sent this to mass spec analysis. Now, here we also see a very significant effect in the Sphingolipid, but it's very different from what we saw in the synthase. In the synthase, we saw a decrease in sphinganin and sphingosine, and we couldn't detect the elevation in the dish, but only in vivo. Here we saw a very dramatic elevation and significant 3 hours post-infection in both sphinganin and sphingosine and 3 ketosphinganin, and also in ceramide, hexosylceramide. Like I marked here in this line all the sphingolipid which were upregulated upon SARS-CoV-2 infection. And when we treat the cells with glucosylceramide synthase inhibitor, we do see that it doesn't allow this increase in hexosylceramide, in GA2, GA1, and as expected. So then we wanted to see again if we see this elevation also in vivo in mice. So we infected K18 humanized 2 mice with SARS-CoV-2, and we treated 5 mice, and 5 days post-infection. In this time point, we took their serum to measure. Now, interestingly, we took 2 mice that had no disease, no phenotype at all. They didn't show any decrease in weight, so this can resemble to asymptomatic patient. And 3 mice that were that have a very severe disease and died from the disease. And we expected to see differences in their lipid profile. But we was very surprised that there was no difference in this point. All of the mice, no matter if they had no disease at all or if they have a very acute disease, showed this alteration in sphingolipids in the blood. Now, I didn't put here all the sphingolipid, just few of them, but this is published. So if you want to see the complete mass spec analysis, it's online. And this is also what we saw in vitro. We see in the serum a very significant elevation in sphingolipid, and was it very interesting for us, also GM3. Now, if you recall, in synthase, for example, we saw elevation of GM1. Now, what I found in the literature, it's not something that we check by themselves, but by ourselves, but that GM3 is more abundant in the lung, which this is the organ that infected by SARS-CoV-2. And GM1 is more abundant in the brain. So that could be due to the organ and cell type, which is being infected. But I'm saying this with a pinch of salt because we need to remember that in these mice, not all the cell, only K18 expressing cells are infected. So it's not one-to-one as the cells that are really infected in reality, and this needs to be verified on human, of course. What we are now currently thinking about this elevation in sphingolipid upon SARS-CoV-2, it was very interesting for us, and we think that it might be involved in many implications in COVID-19 pathology. That, of course, this need to be checked, but this is something that we are interested in now. So one thing is that we think about is maybe the elevation of sphingolipid can lead in the serum, can lead to generation of autoimmune antibody and to neurological manifestation. And I will explain that. We all know Guillain-Barré syndrome, which this is a syndrome which is caused by antibodies which attack antiganglioside antibodies. Now, for now, the assumption in the field of Guillain-Barré and what is known that Guillain-Barré is caused by Campylobacter jejuni. In bacteria came with their own membrane. And in this bacteria, the ganglioside of it's very similar. They mimic the ganglioside of the host, of human. So when you're getting infected by this bacteria, there is a production against the bacteria antiganglioside, but at the end, this ganglioside can also recognize the host and lead to a neurological manifestation. But we hypothesize, one of the hypotheses that we want to look for it now, virus do not come with their own membrane, but if virus can induce alteration and elevation in the host sphingolipid, maybe this is enough to the host to trigger a self-autoimmune anti-ganglioside antibodies. And maybe this ganglioside can lead eventually to neurological or chronic neurological manifestation that we see in viruses, or even they are from unknown etiology. Because as I showed you in the mice, also mice that didn't have any symptoms. It's like a human that don't even know that he was sick or got infected in something, but even though they have an increase in sphingolipid, and if this, and I'm saying if because we're now checking this, and if this is enough to lead to also generation of antiganglioside, it might explain this. Another possible implication of elevation of sphingolipid in the serum could be cardiovascular manifestation. And another thing that is interesting us There are many carrier that carry mutation in gene that is encoding for enzyme which is responsible for sphingolipid degradation. Again, this is one of the topics that I worked in Tony's lab, the lysosomal storage disorder and carrier. So if a non-carrier is being infected by virus, which increases sphingolipid, so there is a balance. It can cope with this, it can degrade them, and he has a mild disease or no symptom at all. But what would happen if we have this increase in sphingolipid in one that has a deficiency in this gene? And he can degrade them much lower, and he has more accumulation. Is that would lead for more severe disease? Is that can be a risk factor, mutation in sphingolipid metabolic pathway that can be a risk factor for virus disease? Again, this is all open question. This is question that bother me at night, and we have no answer for them. And it's just think to food for the thought. So I will just summarize now what we think about the relationship between sphingolipid and viruses. Viruses, early upon their infection, they increase glycosphingolipid levels, which they probably need for the viral replication. Now, exactly why they need them, it's also a very interesting question that we are not fully understanding why they need it. Now, this elevation in glycosphingolipid can alter the host, that there is a viral infection and trigger antiviral immune response. When I say antiviral, it's the type 1 interferon that Tony showed that is triggered. Maybe they can also induce to INKT, another arm of the immune response. And maybe we can take advantage of this alteration in the lipid profile and use this as biomarkers for viral infection on disease. Maybe it can explain some of the long-lasting neurological manifestation, and can it be a risk factor. So this is my our thought now in the lab, and I just want to thank you for listening, and all my group member at the IIBR, especially Roy Abraham, which did most of this work, our sponsorship and collaborator, and thank you.